The title, I apologize to the organizers for having submitted a title that would have disqualified me if it were a competition to actually give a talk, because it was essentially referring to only the very last thing I'm going to talk about. So I decided to give a title that I think was more appropriate. So if you're going to fall asleep during the talk, I will give you a couple of initial ideas of what we're trying to do, and then you can pass out or whatever. But the bottom line is I can tell you that I'm going to be telling you that we have gone from a lab that was mostly working on alternative splicing and mechanistic questions to a lab that actually works mostly on the mechanism of RNA viruses. And then there is a part of the lab which I'm going to tell you about today. And I haven't spoken about this in a while in front of the EURASNET audience, that it's actually using RNA splicing as a tool to try to, and I want to emphasize, to try to analyze the behavior of cancer cells. All of the work I'm going to talk about today was actually done at Duke University in Durham by this group. So I am required by Duke University to make the following statement, and I actually have to say it. I am a founder and have significant equity stake, which could be like $5, if it tanks, in something called Singapore Advanced Biologics. For most of you, this is totally irrelevant, but there's a couple of people here from Singapore, so I have to be careful because we do sell reagents to laboratories in Singapore, and I have to say this. I apologize. I will give you a very quick introduction of alternative splicing of FGFR2 transcripts and epithelial mesenchymal transitions, really in 5 minutes. I will tell you about our tools that we and many others in the field, including Chris Smith here, Masa Higuera, developed to visualize alternative splicing in vivo. Actually, I think very soon, if not already, there's going to be a very nice paper from Brent Gravely using imaging methods to identify, individual cell decisions. I'm going to then go through what we think are three lessons that we've learned, or we're trying to learn, about cancer biology based on these tools. One is all essentially the work of Sebastian Olten when he was in the lab, and I give him a lot of credit for spearheading that work, and in terms of revealing epithelial plasticity in cancer. And then two ongoing stories which have gotten me into some hot water in the field of cancer biology, and that is migration of epithelial cells and several ways of actually achieving metastasis. So first, the alternative splicing of FGFR2, which is what started this whole project, and epithelial mesenchymal transitions. And this is what I'm going to show you here. This is the schematic that was drawn by Eric Wagner many years ago in the lab, based on work that he did and work that Russ Karstens and many other people in the lab and many others in the field, like the Brethnak lab, et cetera. The FGFR2, this is a high-resolution structure, as you can see, of the FGFR2 receptor. It has 3 immunoglobulin-like domains and intracellular kinase domains. And all of the action that we focus on is in this third immunoglobulin-like domain, which actually comes in 2 flavors, and those 2 flavors are given by the functionally mutually exclusive splicing of exon 3B and exon 3C. They essentially, by doing this alternative splicing and providing 2 isoforms, you essentially create 2 different receptors because they have completely different ligand binding specificity. Moreover, they have been shown to be expressed in a cell type specific fashion. So exon 3B is included in epithelium, and exon 3C is included in mesenchyme. And this rule holds for mostly every example we've looked at, except a couple, and you'll see, I'll indicate that we think there are very special cells that sometimes go both ways. But the bottom line is that this, not only is it biologically been shown as a biologically exquisite regulation between epithelial cells and mesenchymal cells, but the function of these isoforms is critical. 3B is required for epithelialization of organs, and 3C, mutations in 3C cause mesenchymal or skeletal abnormalities in both animals and unfortunately in humans. So it is this alternative splicing regulation that we studied, and that was the basis for developing a whole series of tools. The system, the biological system that we applied it to is cancer progression. And this is from a a schematic that Jan-Paul theory divided, and I just want to briefly go through a few steps highlighting the so-called epithelial plasticity or epithelial mesenchymal transitions and the reverse mesenchymal-epithelial transitions. Essentially, this is essentially what happens in carcinomas, cancers that develop from the epithelial layer of organs. You get multiple steps, and the cells start getting disorganized. And even here, you could argue there's already a transition away from polarized, highly structured and well-behaved epithelium into more of a disorganized bundle of cells in carcinoma in situ. Some people consider, including our lab, that EMT has already started here and already started happening here. And actually, molecular markers support that idea. A more frank epithelial mesenchymal transition occurs when cells now become highly invasive. And these cells now express many genes that are mesenchymal in nature. For example, vimentin, and they stop expressing genes that are typically epithelial in nature, like cadherin. These cells are the cells that are presumed, both from pathological studies and studies of Robert Weinberg, and also mechanistically, that the ones that invade through. And here, Jan-Paul in this slide put them as being the ones in circulation. And then they have, so in metastasis, not only do you have to intravasate into the blood supply, but now you have to get out of it and seed or colonize other sites. And the prediction originally from a German pathologist called Thomas Brablitz was that because of what he had observed in human colorectal cancers, the prediction was that there's going to be a reverse mesenchymal to epithelial transition at that point. So in the talk today, I am going to highlight three aspects of this in which I think our reporters have given us some insights into what's going on. The first is during primary tumor formation and the ability of cells to actually undergo these plastic transformation, perhaps something we did not expect before we observed it. Second is some insights we've gotten into the ability of cells to migrate and whether you require EMT to do this. And then finally, we're actually addressing the question, do you require mesenchymal epithelial transitions to actually form macrometastasis, real colonies in the distant tissues? So to move forward, let me tell you, I'm going to spend 5 minutes or so describing tools we've created, alternative splicing reporter tools, to actually try to address some of these questions. And I'm going to go a little bit historical because I'm going to show a slide that I showed for the first time at a pre-EURASNET meeting in Oslo. And it was a proof of principle idea that we could actually, based on mechanistic studies of Russ when he was in the lab, of Eric Wagner, Vivian Bonanno and Sebastian Altian and others in the lab, created a series of constructs where we can actually image, in a living animal, we could image intron splicing silencer dependent repression of exon 3B. This was actually very exciting for us, but it was really only a proof of principle because these particular constructs that were in these animals were actually not really able to tell us the difference between an epithelial cell and a mesenchymal cell. They merely reported on the activity of the silencer and presumably on proteins of the PTB family. Now you can see that just to give you a little bit of background on the system, because we're going to come back to this system briefly later. What we did here is we actually created transgenic mice where one reporter, which is a constitutive reporter, was in one allele of the ROSA26 locus, and the other reporter that actually reported on the splicing of 3 was in the other allele. So these animals are always heterozygotes at the ROSA26 locus, and one reporter controls for the other. So you can see here that in the eyes, which end up, these were neurons in the retina actually when we dissected them, you can see that the control reporter doesn't change, but here you have very clear repression of exon 3B that depends on the silencer, because this is a wild type construct, and this is one where we had mutated the two silencers that bind PTB. So this proof of principle emboldened us and also deceived us to go ahead and use these constructs, similar ones for cancer, and those have been a lot more difficult to work with in several different ways. But nonetheless, we've had some successes. And the system that we decided to use was the cancer progression in prostate cancer. So I'm going to briefly go through what is considered to be the unfortunate path of prostate cancer when it goes wrong. Most prostate cancers don't, most men, and as we approach a certain age, this is a sensitive subject, but most men will develop prostate cancer. And the majority of those men will actually not have this course, thankfully. But about 25,000 out of the over 150,000 that are diagnosed in the US every year will go through this, which is the cancer will appear, and when it's above this line is when you actually can feel it or can detect it. It will be detected, and usually, depending on the type of tumor, but mostly people will be treated with androgen deprivation because most of these tumors actually require androgen receptor and androgens to grow. The tumors will essentially revert. And interestingly, most of these tumors, when you study them, when you look at them, are epithelial in nature, and they express FGFR2/3b. Between months and years later, after androgen deprivation therapy, the tumors recur. And in the majority of those cases, those tumors that recur have a much more aggressive phenotype. The recurrence are usually not in situ, but rather in distant parts of the body, mostly in bone. And many times, those tumors actually are mesenchymal in nature or have mesenchymal character. And many of them express FGFR2/3c. So there's been an epithelial mesenchymal transition in whatever cells were left behind after the tumor is removed and androgen therapy is instituted. And what we did was we used an animal model system called the Dunning system that was actually pioneered at Hopkins, but then used by Wally McKeon in Houston for many years. And Sebastian Altian, who took over this work, created a series of reporters in collaboration with others in the lab that actually now permitted us to, in many ways, tell the difference between an epithelial and a mesenchymal cell. So the idea was, it's interesting because almost everything I'm going to tell you in this small vignette of Sebastian's work was a result of the control in an experiment that globally failed. And that was mostly because we actually wanted to recapitulate that whole process by putting a very benign type, not benign, that's the wrong word, an indolent prostate cancer cell and let it go through all of the process. And that did not work, but Sebastian found some gold in the control, and I'll show you that data. And I'll be brief about that because most of this has been published of this part. So essentially, based on a lot of analysis, mostly from Russ Carstens when he was in the lab, and then later on his own, and of Eric Wagner's and other, we knew that there were a series of important cis-acting elements that were required for epithelial versus mesenchymal inclusion of exon 3C. This exon is the one that goes in mesenchyme, so what we're actually analyzing here is an epithelial cell's efficient repression gives you, in this case, DS-red, or RFP, as I abbreviated here. And in mesenchymal cells, the exon gets efficiently included. And the first thing to do was to actually use the system that I mentioned. This is the DT and AT3 Dunning cells, and they actually come from each other. The AT3, this arose spontaneously in the prostate of a rat, and this is a post-EMT tumor that actually emerged after these tumors grew for a while in castrated rats, in castrated male rats, or in female rats. There were several lines that were developed by this reimplantation in vivo, and this is one of the more aggressive ones, and we refer to this as a post-EMT system. This is just an old gel from Russ Carstens when he was in the lab, and he showed that the FGFR receptor was actually essentially almost 100% spliced one way, the 3B way in DT cells, but the 3C way. And we could see the same thing with molecular reporters. And further work from Sebastian and others showed, essentially did sequencing of the transcriptome, and these cells clearly have an epithelial signature. These cells have a mesenchymal signature, and also a more stem-like signature, which is not surprising at all, given some of the of their aggressiveness. I also want to say that when we actually compare it using gene set enrichment analysis, these cells are much more like the cells that in humans are metastasizing in prostate cancer. Okay, so using the system, these cell lines which grow in culture and grow in animals, although the DTs, as I said to you, as I alluded to, the DTs take months, if ever, to give you a tumor once in so many rats, whereas the ATs actually can grow very, very well both in culture and also in tumors. But in tissue culture, the reporters behaved as we expected. I'm only showing you the fluorescence, but obviously we tested the molecular, the RNAs, and what you see is in epithelial cells, the exon is repressed, which we could see by RT-PCR, and then you actually get fluorescence. In mesenchymal cells, the control here, which is constitutively splicing, gives you fluorescence, but this reporter now includes 3C efficiently and blocks RFP. And again, this was confirmed at the level of RNA. So the big surprise with this came later. I just want to show you here the tools and then explain to you about a couple of new tools we've developed that are based on these. So this was very robust in terms of, and gave us maybe 10 to 15-fold discrimination if you actually quantify. The data look absolute, but it's really a 10 to 15-fold discrimination. But we wanted to actually express a series of enzymes rather than, I guess you could consider fluorescent proteins. They're really not enzymes. So we wanted to express enzymes under the same control. In order to do that, we wanted to have a larger than 10 to 15-fold discrimination. So several individuals in the lab, including Matt Marengo and Tristan Babler, used constructs that Sebastian had already made, and then they altered them in different ways. And we have a whole series of these, which we call synthetic constructs. And I'm going to give you just 2 examples and then mention a few of them later in the talk. So essentially, what they do is they do something that I think Ben Blenkow and others have taught me doesn't really happen a lot in nature, which is that the same genes don't get regulated by multiple different ways, like transcription and splicing seem to be orthogonal. But I've always been interested in those genes that actually are regulated by both mechanisms. And that's what we did. We synthetically made these genes now regulated by transcription. In this particular example, it's an EC-adherent promoter, which is 5 to 10 times higher in epithelial cells than it is in mesenchymal cells, and coupled it with a splicing regulation. So I don't want to go through all the details here. This was done with luciferase, and I'll show you later that we've used similar constructs with 2 other enzymes, Cre recombinase and diphtheria toxin. And what can be seen from these data, and it has been repeated in a whole series of different systems, so this is from the DT83 system, is that when you combine splicing and transcription, we end up getting between 50 and 60-fold discrimination between mesenchymal cells. This is a combination construct here, and epithelial cells. But something else was even more important, and that is that the threshold expression was much lower. So for experiments like I'm going to show you with the diphtheria toxin, this was actually very critical. Not only we wanted to have discrimination between the two, but we also wanted to be below a certain threshold of expression to be able to actually use them as selectable killing agents or marking agents. So this has been a nice move forward, and I'm going to show you that we have borrowing a young, very talented medical student from Masa Hagiwara's lab who's been on loan in the lab for a few months. We now have developed another wrinkle in this, which is to add microRNA sites. Not surprisingly, you can then also add that to the regulation. And here we have now in this system, which is an epithelial versus mesenchymal, but in particularly there's a whole series of epithelial cells that have very high MIR200 levels. And there's many mesenchymal cells that have very low MIR200. So we can actually regulate the expression of GFP here by an epithelial, by a mesenchymal specific promoter, in this case by mentin, and also include in the 3' UTR the microRNAs. And the promoter actually gave us very little discrimination in this system. Maybe because MCF7 cells are not 100% epithelial, they're actually already transformed. But the MIR gave us very good regulation, and now we're actually adding to these the alternative splicing and see how much regulation we can get. So these are the sort of tools that I'm going to show you, and I think before I go to my conclusion from this part, I think it's actually very appropriate to discuss reporters in this context, given this paper almost 30 years ago from Tito Barale and Alberto Gombrich and Karen V. Pedersen. that actually was, at least for me, one of the things that inspired me to use reporters in cells rather than doing in vitro experiments. So Tito, thank you for many years ago inspiration of this. I have to admit that it was Alberto who first pointed this out to me, but that's okay. So the conclusion is that FGFR splicing reporters visualize cell type specific regulation, and in particular we have reporters that we believe give us identify epithelial versus mesenchymal phenotypes. And these synthetic reporters provide the synthetic reporters that actually multiply or different regulatory elements like transcription and splicing give us better differential. But that was all tool building, and then the question I think that one has to address with some sobriety is, have we learned anything from them? And I'm going to try to convince you that we have. I'm also going to tell you that we're not all the way there. So the first question that we asked actually came, as I said, from experiments that Sebastian did, and then it came out of a control. So I cannot take credit for having asked the intelligent question and said, oh, we got the answer. But rather, I remember the first time I dissected one of these rats. Actually, it was right before Sebastian got into the lab. We did a proof of concept experiment. And I said, this is completely, and I used a word that I can't repeat here, but it involved several expletives. I said, this is wrong. Because these cells were mesenchymal cells, were not supposed to be giving you a signal, and there was some signal there. So Sebastian was just joining the lab, and I said, Sebastian, you need to do something about this. So he did some very careful experiments, and I'm going to just give you the highlights. And essentially, these 83 cells that I told you, they're post-EMT. They have a very highly mesenchymal expression of mesenchymal markers, very high vimentin, very low cadherin, very high twist, one of the master regulators of mesenchymal behavior. And as expected in that fluorescent reporter I showed you before, they should be dark, because the 3C exon is getting included, is interrupting the RFP. But in almost every single tumor that Sebastian got, out of looking at many rats, he found things like this. And he went on to show, and this is not a coincidence, this was actually right at the edge of where, these tumors can be very easily dissectable, and this was right at the edge of the tumor and the normal tissue. Sebastian went on to show that these islands of fluorescence co-stain with many epithelial markers and also correlated with areas of the tumor that had started losing staining of vimentin. And our conclusion, which has now been confirmed multiple times, is that these islands represent a transition back to a more epithelial state. I'm not going to say these cells are epithelial, but they clearly express epithelial markers. So they've transitioned somewhere back. They've gone through what we call or what's called in the field, mesenchymal to epithelial transition. And what this said to us is there was plasticity even within the primary tumor, which was surprising for such an aggressive cancer as AT3 is a model for. And then this also inspired us, and I still remember the meeting we had with the GU clinicians where Sebastian and I went to see them, and we said, you know, this could mean that we may be able to find similar things in humans with very aggressive cancers. And at that meeting, we suggested that the place to find this was probably the circulation. There are circulating tumor cells in patients that have metastatic disease. These circulating tumor cells are presumed, but have never been proven to be, the sources, the origins of metastasis, of distant metastasis. And about 60% of prostate cancer patients with what's called castration-resistant prostate cancer have these cells in the circulation. And one of the things that we did in collaboration with Andy Armstrong and a whole team of clinicians in a series of clinical studies is we showed that indeed we could find cells in the circulation that were highly plastic. And they're highly plastic because, as is exemplified here, this is a cell that we know is a cancer cell because it went through a series of parameters of checking. First of all, it is actually purified with an FDA-approved protocol for getting circulating cancer cells, and it expresses both E and N cadherin, markers of two sides of the EMT barrier. Whereas other ones only express N cadherin, for example, or express E cadherin. But even this cell, we would argue, is actually plastic because it is captured with an epithelial ferrofluid. The next slide just essentially suggests from these clinical data that this is not a rare phenomenon. The majority of the patients and the majority of the cells within the patients suggested plasticity. And now we have a collaboration with Illumina where we're actually sequencing very low numbers of these CTCs and trying to develop a signature for epithelial versus mesenchymal plasticity. However, this was, at least for us, validation that what we had seen in animals was actually happening in humans. So what we saw in animals was this post-EMT83 system revealed plasticity, and we predicted it would be there in human cells. I think we got a little bit lucky, for reasons I'll tell you in a moment, that the prediction actually ended up being right. And I have to give Sebastian a lot of credit for having suggested that we look at CTCs. His MD degree was more valuable than my MD degree, so because I forgot everything I learned in medical school. But so then we had done this, and then we actually decided to ask a couple of other questions about this. And the first one I'm going to just spend a couple of minutes on, because I think I only have like another 6 or 7 minutes if we. And that is, we got a surprising result about migration of epithelial cells. Let me frame this with the dogma. The dogma, if you look at people who really understand migration, like John Candelas, would never agree to this. But if you actually look at the cancer biology field, and you actually look at the tables where they describe epithelial versus mesenchymal transition, versus mesenchymal cells, one of the things that you always see in the column under mesenchymals is migration, and then you see epithelial cells as non-motile. Well, I think that's complete bunk. Candelas agrees, but even people who have published data like this don't talk about this in the literature. So Jason Somarelli made another mouse, and this mouse is very, very similar to the first one, and again, allelic regulation. So we have in one allele of ROSA, we have now we flipped the colors. Sorry about that, but we flipped for a good reason. We had to use different colors here. It has one allele, the control is red, and then the experimental is green. But these are now the ones that distinguish between epithelial and mesenchymal cells. So the first thing Jason Somarelli did was he showed that indeed in these animals and in tissues extracted from these animals, we actually got epithelial specific. For example, you get epidermis but not dermis. You get keratinocytes expressing but not fibroblasts from the skin expressing green. Unfortunately for us, the penetrance of the signal was not full, so we haven't been able to look at all the tissues. We've had to essentially concentrate on the skin in these animals. We don't understand why that is. They're exactly in the same location as the previous ones, but they're much more complex reporters. But nonetheless, for skin experiments, they've been very useful. And one of the things that we decided to do is take a piece of skin and do a wound healing ex vivo. So you can actually take a skin explant. I don't want to get into the details because it's pretty nasty. But you get skin explants from these animals, and you actually look at wound healing and migration of cells in ex vivo in dishes. And I'm no microscopist, but I'm going to show you a few seconds of a film that suggested to us that cells could migrate while not having to undergo an epithelial mesenchymal transition. And this is only a suggestion. I think there are some caveats to this, and that's why we then did a whole series of experiments. But this is the edge of an explant right there from the skin of some of these animals. We did this with both control, and we have a control animal that has mutations, but all the data I'm going to show you is from the animal where if you see fluorescence here, that means that the cell is epithelial, or at least was epithelial before the GFP decayed. And that's one of the caveats here, but this is several days. So the bottom line is, I'm going to show you just if it works. Let's see. It worked perfectly well yesterday. And there we go. So those are cells coming out of the explant, and you'll see them moving, and you'll see them dividing also. Okay? So the last thing I want to tell you is one last thing. So this is work in progress, and that is, instead of actually marking the cells at the moment that you're looking at them, to actually ask, can we ask the question, did the cell or any of its ancestors ever went through MET? In other words, these mesenchymal post-EMT cells, have any of them ever, so it's lineage tracing, or the other reporter is, if the cells undergo MET, will kill them. Okay? So those are the general parameters. So the first experiment is a lineage tracing. None of this last few slides have been published, and I know they will be broadcast in the video, and I will, send some of my friends from New Jersey after people if they ever use this. No, I'm kidding. But so here, this is a reporter. The only thing I have to say about it is the logic is the same as I showed you with the luciferase synthetic. It has an ECADherent promoter, so it'll be expressed primarily in epithelial cells. Has a 3C exon, so again, in mesenchymal cells, it will interrupt the ORF. And the ORF in this case is Cre recombinase. In the next case I'm going to show you is diphtheria toxin. So in epithelial cells, you express Cre expression. In mesenchymal cells, you don't. These cells have been previously engineered to contain a reporter for Cre recombinase. So before recombination, they're red. After Cre recombination, they're green. There are multiple insertion sites, and not all of them convert in every case. That's something as a caveat in the back of our minds. But the controls work. These are now in tumors. We've done this in culture. The controls work very well. If you actually put a totally rampant Cre recombinase, you get complete conversion to green. If you actually put this one driven by both transcription and splicing, you only get very small amounts of the tumors to show green. And then the question was, do we get a higher preponderance of these cells in metastasis than in the tumors? And the answer was not significantly different. So what it suggested to us is this is what happens in culture in several weeks of growth, which is you actually have a certain percentage of the cells flipping over to expressing, to being green. We interpret that as having undergone MET at one point or another. This is what happens in primary tumors. And this is what happens in metastasis. So I want you to realize that this is percentage of the events, because we can count all the events as events that are red versus green. So there's a lot more variability in the metastasis, but that means that 95% of the events, those cells never had an ancestor or themselves never underwent MET. So what this suggests to us is that the requirement for MET to create these metastases in the lung is actually very limited. So it may be that in some cases it helps, but the majority of the metastases that we find in the lungs do not have to have developed from cells that went through MET. It's an indirect argument, but I think together with this next one, it's starting to suggest to us very clearly that this is not clear. And here again, it's very simple. Now you kill cells that undergo MET, right? These are AT3 cells, so we can kill epithelial cells very easily. The AT3 cells, we get many survivors. They grow up, they're expressing the reporter, they're repressing, they're including 3C very well, so very, very low levels of diphtheria toxin are being made. And then we ask the question, if we actually put them, do they form tumors? And the answer is yes. And in this experiment, we had to expedite the metastatic process, so we injected into the tail vein of these nude rats. This is all done in either syngeneic rats or nude rats. And we found no difference between let me say one last thing. All of these cells have a GFP marker in them anyway. So we found no difference in the number of metastases, again suggesting that the transition back to an epithelial state was not required. So to summarize, we believe that this lineage tracing and suicide reporters suggest that metastatic colonization does not require MET. And this actually fits a model of Thomas Brablett's, where he thinks that there are two types of cancers that actually metastasize through very different processes. And he claims, and I think he's probably right, that those have very different clinical implications. So we believe AT3 cells are a model of this undifferentiated type of cell line that doesn't have to go through MET to form macrometastasis to colonize tissues. This is only half of the experiment, because we're actually doing this now with several different human cell lines that are more traditional. We actually believe that this represents a model of something called carcinosarcoma, which is very rare. It's only 2 to 3% of prostate cancers, but they're extremely lethal. And we think that that's what we have actually been observing, and now we're using more conventional models of prostate adenocarcinoma to actually test these ideas. So I told you about 3 different places in the cancer biology or cancer progression where I believe our splicing reporters are giving us a contribution, albeit an incomplete one yet, but it's giving us shedding some light in some of these questions that have been more difficult to deal with, particularly when we combine regulation with transcription. And I want to thank people who did the work. So these are the people in the lab right now, Jason Somarelli and Danine Schaefer, and then these are some of the contributors that throughout the beginning of this project, and I want to give particular thanks to Sebastian Altian and Vivian Borano for having done this, and then some very good collaborators, both at Duke and at Hopkins and at UCSF, and the sources of funding, and I thank you for your attention.